Studying the effect of an enzyme is difficult because the absence of an enzyme may mean that no reaction will occur. The concentration of enzyme, substrate, temperature, and pH can affect the performance of the enzyme as well as the rate of reaction. Enzyme assays are also known as enzyme studies, and they're used to essentially simulate biological conditions and emulate the natural environment of the enzymes in order to perform tests on them. Enzyme activity can be measured by the decrease in concentration of reactants or increase in concentration of products, generally mole per decimeter cubed per second, and it's essentially the same way that you would find any other rate of reaction. So the effective concentration is the first effect. The rate of reaction is compared by analyzing the initial rate of product formation over time. In a graph of concentration versus time, the rate of reaction is the slope of the tangent line near the beginning of the reaction. Concentration of the enzyme will directly affect the rate of chemical reaction because there's more enzyme to catalyze the reaction. The concentration of substrate, however, concentration of substrate affects the reaction in a different way. When the substrate concentration is relatively lower than the concentration of the enzyme, the rate of reaction will increase as the substrate concentration approaches the level of concentration of enzyme. Concentration of substrate. When enzyme concentration is relatively lower than the concentration of the enzyme, a rate of reaction will increase as substrate concentration approaches the level of concentration of the enzyme. When the substrate concentration is relatively lower than the concentration of the enzyme, the rate of reaction will increase as substrate concentration approaches the level of concentration of the enzyme. If the substrate concentration is relatively higher than the concentration of the enzyme, there is not enough enzyme to catalyze the reaction, so the rate of reaction approaches a maximum. In the middle, between the two extremes, when all active sites are engaged in catalysis, the maximum rate of reaction is achieved. Temperature affects the rate of reaction by generally increasing the rate of reaction as temperature increases. Because enzymes are globular proteins, heat can easily disrupt the intermolecular forces, causing enzymes to denature, or essentially change shape. A denatured enzyme cannot perform its function since shapes of active sites will be different. Many organisms have temperature control systems to maintain body temperatures, so enzymes can work optimally. In the graph, you can see the sharp increase of the rate of reaction as the temperature increases to a certain point. And then after that, there's a sharp drop because the enzyme has essentially been denatured. The increase is caused by the rate of reaction, the number of collisions increasing as the temperature increases. An example of the scenario or the situation would be gelatin, which is used in cooking to add thicker texture to a material such as jelly. Some fruits contain enzymes, however, called proteases that can break down gelatin, and which means that they can't really be used in jelly. However, canned fruits that are heat sealed can be used since the enzymes that would normally break down the gelatin have now been denatured, and the protease enzymes are inactive. The effective pH can also affect the rate of reaction. Changes in pH can cause denaturation of the enzyme, and for many enzymes, the pH must be close to neutral. For others, however, for example, those in the stomach used to aid digestion, must perform pH ranges outside of neutral. Significant changes in pH can cause amino acid residues with acid-base activity to gain or lose protons, changing the shape and interactions with other amino acid residues. The pH of aqueous environments can be maintained by the action of a buffer system, which we learned in class. And here we can see the effect of acidity on the reaction rate with pepsin, which is a protein that operates at a very low pH and is optimally placed at around pH of 2, as compared to the salivary amylase, which tends to peak in its ability to affect the reaction rate at about 7. Lastly, we have the effect of heavy metal ions. some heavy metal ions have the ability to bind to an enzyme, like non-competitive inhibitors, causing a decrease in the activity of the enzyme. Common heavy metal ions include lead-2 and mercury-2, which bind to a variety of enzymes, causing long-term and short-term health effects. For example, lead and mercury ions are both neurotoxins, since they can cause permanent brain damage. Silver, lead, mercury, and copper ions can form a bond with thiol, which is an SH group, on cysteine amino acid residues, and the presence causes a change in the shape of the enzyme, which results in a decrease of function. Since the bond is covalent, the binding of a heavy metal to the enzyme is strong and permanent. And as we can see, the change in shape of the enzyme actually causes the active site to change, which affects its ability to act as an enzyme.